Okay, welcome to week 9 in the molecular epidemiology course. And this week and for the next couple of weeks, we are going to be focusing on the molecular epidemiology of cancer. And so tonight we're just going to do an overview of the molecular epi of cancer and cancer biomarkers. And in the week 9 weekly session in the course, you have an assignment this week, which is a great review article that discusses molecular epidemiology of cancer and goes into some detail on specific biomarkers with certain types of cancers. So we'll start out, what is cancer? And cancer in the most basic terms is uncontrolled cellular growth. So typically you'll have some cells, our cells in our body, they divide, multiply, cells get old when they're past their prime, they die off, new cells take their place. When you have cells that just start to grow uncontrollably and don't die, and you get this massive overgrowth, that is when cancer happens. And typically these mass of cells will form a tumor. And there are two different types of tumors. There are benign tumors and there are cancerous tumors. So benign tumors are a cluster of cells that typically will not spread or metastasize, and they are not considered cancerous. So those can typically be removed if they were to get large enough, and there wouldn't be any risk of them spreading. However, cancerous tumor, there's many other things that would need to be done. So for a cancerous tumor, again, you could have surgery to remove that tumor, but that wouldn't usually be enough because there could be a risk of some cancer cells that weren't removed that can then spread. So even if you were to have surgery, usually there would need to be some chemotherapy in order to kill any cells that remained. Now, the main cancer types, when we're referring to all of various cancers, they typically fall into one of these 4 cancer types. The most common being carcinomas. There's also sarcomas, leukemias, and lymphomas. So for carcinomas, these usually are in the form of solid tumors. They typically begin in the skin or in the coverings of organs, so the tissues that line organs. Most common types of carcinomas: prostate cancer, breast cancer, this is lung cancer, I've got a typo there, and colorectal cancer. So I will fix that slide. So that's lung cancer. Sarcomas begin in tissues that support and connect. So typically you'll find sarcomas in fat, muscle, nerves, tendons, joints, blood vessels, cartilage, and bone. Leukemias are cancers of the blood cells, and there's 4 main types of leukemias: acute lymphocytic leukemia, chronic lymphocytic leukemia, acute myeloid leukemia, and chronic myeloid leukemia. And then there's the lymphomas, and these are cancers that begin in the lymphatic system, and there are 2 main types of lymphomas: Hodgkin's lymphoma and non-Hodgkin lymphoma. So cancer spreading. So typically you'll have your cancer cells that get into the blood or the lymph system. And when they get into the blood and lymph system, those systems can carry those cells anywhere in the body. And so when you have spread from one area to another area, that is what's termed metastasis, where you have the spread of the cancer. So common spread of cancers is in the lymph nodes. So typically you will have lymph nodes surrounding a tumor that will have some cancerous cells. So typically if surgery is done, the tumor is removed as well as all of the surrounding lymph nodes to remove as much of the cancer as possible so that you reduce the risk of it spreading even further. If it gets into the bloodstream, it can spread to very distant areas, so not just the surrounding lymph nodes, but any other organs in the body. So one thing to remember is regardless of where cancer spreads, it's named for the location where it begins. So for example, if an individual has breast cancer and that's where the tumor started and it metastasizes and it's found in the lung as well as in the bone, for example, that particular cancer, even though it's in the lung and the bone, it is not lung cancer and bone cancer. It is still breast cancer. It's just metastatic breast cancer. So the cells, those cancer cells, originated in the breast. So that is why you would typically find someone that would have a certain type of cancer, but you know that they have cancer in other organs. So cancer genetics. So typically what happens with these cells is there are mutations that occur, and those mutations are what cause those cells to overgrow. So the two types of mutations are acquired mutations and germline mutations. So most cancers arise from acquired mutations, and there are environmental factors that will increase a person's risk for acquiring mutations that can lead to cancers. And these include ultraviolet light, viruses, enhanced age, tobacco use, alcohol use, not eating healthy, all these types of environmental factors. Now, germline mutations are mutations that occur in the sperm cells or egg cells. So these types of mutations are inherited. They can be passed on directly to offspring. So acquired mutations will give you sporadic cancers, whereas germline mutations will lead to inherited cancers. Now, germline or inherited cancers are less common. 5 to 20% of all cancers are inherited cancers. Now, there are many different types of genes and mutations within these genes that are linked to cancer. And the article that we have this week goes through a lot of these different genes, which have now become biomarkers for various cancers. So some of the common genes that have been linked to cancer are tumor suppressor genes. So of course, if you have genes that suppress tumors, that there's a mutation and they no longer are suppressing, you can get tumors that develop. Oncogenes, genes that lead to cancers, and a common type is DNA repair genes. So in our bodies, we have mutations that occur all the time when our cells are replicating, and we have DNA repair mechanisms that will go in and fix those mutations. However, if those mutations are not getting fixed, that could have deleterious outcomes. So tumor suppressor genes limit cell growth, they repair mismatched DNA, they control cellular death, and so tumor suppressor genes are critical. And so when you have issues with these genes, you have a high risk of cancer. So examples of these are BRCA1, BRCA2 involved in breast cancers, P53. P53 is involved in many different types of cancers, and TP53. And more than 50% of cancers involve P53 gene mutations. So P53 is critical. Now, oncogenes, you can get acquired mutations that cause a cell to become cancerous. And there are two oncogenes that are common, and these are HER2. HER2 is a protein that controls cancer growth and spread, found in some cancer cells, such as breast and ovarian cancer cells. So many times with breast and ovarian cancers, you will hear them referred to as HER2 positive or HER2 negative. There's also the RAS family, which is a large family of proteins that's involved in cell communication, cellular growth, and cell death. So whenever you have issues with communication, growth, and death, that can lead to cancers. Now, we mentioned DNA repair genes, and these fix any mistakes that are made when our DNA is copied during cellular replication. Many of these DNA repair genes are tumor suppressor genes. So that makes sense because the tumor suppressor genes, of course, are involved in cancers, and whenever you have mutations and they're not getting repaired by these DNA repair genes, you can lead to issues. So examples, again, BRCA1, BRCA2, the notorious P53, and mutations in these repair genes can be either inherited or acquired. Now, the issue with studying cancer, and if these are issues that can be the same with other types of diseases, are that there are tons of different cancers out there, and there are cancers that have not been to date linked to any specific genes. Also, like some other diseases, many different gene mutations could be involved in cancer. So you can't really just look at this one gene mutation. You have to really, it's much more complicated. You have to look at multiple gene mutations and trying to figure out what role each of them play in cancer development. You also have environmental factors. So, smoking has been shown to be highly associated with cancers such as lung cancer, many cancers, but one very high association is lung cancer. So again, what is the main cause of someone getting a cancer? Is it, an inherited gene? Is it a combination of multiple acquired mutations? Or is it all of those plus environmental factors? So it does make studying these types of diseases incredibly challenging. But the fact that we do have genes, we have associations of environmental factors, so we do have an enhanced understanding of how certain cancers can develop, what biomarkers and mutations do we know, which allows us to detect cancers much earlier than we used to. And a lot of these genes also are the targets of new therapies that have been developed. So typically with cancers, the treatment is surgery, radiation, and chemotherapy. But now we actually have new therapies, these targeted therapies and immunological therapies that go after these proteins and, these immunological factors that are involved in cancers, and they have much fewer side effects than, for example, chemotherapy. Chemotherapy are highly toxic chemicals that kill cancerous cells, but also kill healthy cells. So that's why chemotherapies have a lot of side effects, because you're killing off healthy cells, you're killing off your white blood cells, you're putting individuals at, making them severely immunocompromised by getting rid of a lot of their white blood cells. It can kill off red blood cells and make people incredibly anemic. Radiation is more targeted than chemotherapy, which is going through your bloodstream and getting through all of your body. But radiation is targeted, but again, it's killing all of the cells in that surrounding area, including healthy cells. So there's a lot of side effects. So these new targeted therapies are very promising. And so that is the focus of a lot of the more recent literature in cancer biology. So, like I said, this week we have a paper, and in this paper that you're going to be reading, it goes through a lot of these molecular biomarkers and a lot of these genes and proteins that are associated with cancers. So with these molecular markers, they help differentiate tumors, because you can have tumors that, for example, you can have tumors in one area and it spreads to another area. So for example, there's cases where individuals will have cancer and it's identified, for example, in their liver. They happen to get a scan and there's some tumors noticed in their liver, so they get a biopsy done of their liver. But when they look at the cells, in the cancer biomarkers in the liver biopsy, they realize that cancer is actually maybe not liver cancer, but it's lung cancer. And then sure enough, when they go look at the lung, yep, there are tumors there. So it originated as lung cancer, it spread to the liver. And you can differentiate tumors based on these biomarkers. And some even in the same area or the same cancer in two different people, histologically, when you take a biopsy and just stain the tissue and look at it, it could look the same. But when you actually look into the molecular, go deeper into the molecular differences between two different tumors, you actually can see that they are different. And so when two different individuals have the same type of cancer, but they actually are different subtypes, there could be different treatments, even though it's the same cancer, but different treatments for each individual's cancer subtype that might be better for them. So looking at these things at the molecular level is very, very beneficial. So like we just said, responses to treatment could be determined by the molecular subtypes. Certain, it's being found that, certain types of mutations respond much better to specific medications or respond worse to specific medications. So why put someone, for example, through a chemotherapy that is not actually going to be as effective against that particular subtype of cancer? So molecular is playing a very large role in cancer biology and looking at the epidemiology and the associations between specific mutations, environmental factors, and cancers is, just a very large field of study. So epi on its own has been incredibly successful identifying environmental factors and things like that that increase an individual's risk for cancer and have helped develop some good prevention strategies. Stop smoking so that you lower your risk for lung cancer, for example. Now, the etiology of cancers is still poorly understood. There's still a lot we need to learn. We still don't know, like we said, not every single cancer is associated with specific genes or mutations. So with traditional epidemiology, we have learned a lot using questionnaires, interviewing individuals, have you ever smoked? Have you ever drank? How much? What do you eat? Do you eat foods high in fat? Things like that, to make those associations between specific types of environmental factors and potentially having a higher risk for cancers. But when you add molecular into it and look at things at the molecular and cellular level, it does provide some new insights. So that is what we're going to be focusing on over the next couple of weeks in the course. I think you'll find the article for this week very enlightening as to some of this very specific genes and proteins and mutations that are involved in various cancers. And some of these genes and proteins are the same in different cancers. Now, in order to do the article in the assignment this week well, you will have to do some extended research in order to answer some of the questions. So by doing some additional research in the literature, it's going to enhance your understanding and identifying some additional risk factors, additional biomarkers, and roles that these genes and proteins are playing in each of the different types of cancers. So I think once you read the article, and there's also a video this week that you should watch, really short video, and answer all the questions in the article, I think you'll find it quite informational. But I'm going to close there, and I'm going to open it up. There are a couple of you here tonight, and see if you have any questions on molecular epi of cancer, which we're just starting to talk about, or on the course so far, or on your midterm project, which will all of the different sections will be graded in the next couple of days. But hopefully you were able to learn a lot in doing your midterm project and in doing your peer reviews, so you can learn not only what you did your research on, but what all of your classmates also did their research on. So anybody have any specific questions?
I actually have a question about the assignment for this week.
Sure.
On the weekly sessions list, it says week 9 assignment, and it says read the article, and it has all the questions. Then at the bottom it says answer by 11:59 on Friday and respond to a post by Sunday, but then the link for it just links to like the regular submit an individual assignment type of link, and there's no discussion board post. So
it should be a discussion. Let me go in and fix that. Yeah, it should be a discussion, so that yeah.
Okay, that makes sense. Thank you.
Great, thank you for letting me know that. Anyone else have any other questions? If not, well, you can go back to your evening and maybe since you might have a few extra minutes, since we didn't take that long in the short lecture, you can have time to watch the video that's posted in the weekly session and start reading the article for this week so you can answer the questions by Friday.
Thank you.
Thanks. Thanks for coming.
Thanks. Bye.
Bye, everybody.